Feynman also suggested that really understanding and manipulating things on a small scale might enable some real revolutions in the field of materials. And this, I think, is really the sort of forefront of where most research in nanotechnology is today. It's the re-engineering of materials to do new and different things. And that takes two sort of different forms. Sometimes it's actually building new materials, designing or discovering molecules that have geometries that we haven't seen before. A good example of that is this thing called the carbon nanotube. This is a long, thin tube of carbon atoms that can have some really amazing properties. They can be super strong or extremely conductive. They can be semiconductors. They're extremely flexible. They're good conductors of heat. And they come in many different forms that share some of these many different properties. It's actually a naturally occurring form of carbon, but we've discovered it recently and can manipulate it and make some new molecules out of it. This is a picture of a mat of those tubes all grown together. But sometimes it's also just taking materials that we already know about and making very small pieces of them and finding that they behave in a different way. This photograph is of a material that we call quantum dots. Now, these are very tiny little pieces of a semiconductor material that are grown in solution. So we take a solution of semiconductor and add another material, and the pieces sort of start to grow. They self-assemble up into these little chunks of semiconductor. Now, what's interesting about this photograph is that every one of those vials contains the exact same material. The only difference between this one and this one is the size of those chunks of semiconductor. And that, if you think about it for a minute, is really contrary to our experience, right? You wouldn't expect a jar of big marbles to behave fundamentally differently than a jar of small marbles, and yet that's essentially what's going on here. These are the smallest particles, and these are the largest. It's the same semiconductor material, but just by changing the particle size, we're able to change the way that it reacts, in this case, to ultraviolet light. So that's really the forefront of materials. Sometimes it's designing new materials, and sometimes it's just designing new structures that make old materials behave in a new and different way. And this is really at the forefront of the applications of nanotechnology, and it has applications in some of the most important issues in science and technology that are facing society, like energy production. Lots and lots of people in material science and in nanotechnology are trying to create less expensive and more durable photovoltaics for solar cells. We could, if we had the technology, generate all of the power that we need on this planet directly from the sun with no waste and zero emission. But again, making the panels that we have today is a cost-prohibitive process. How do we have better materials to make more panels? That's a pressing question. The other great resource question in the world, obviously, is access to water. A huge swaths of the developing world, and in some places the developed world, don't have enough access to fresh water, and that problem is expected to worsen as the climate changes. Now, there's plenty of water on this planet. The surface of the Earth is 3/4 covered with water, but of course we can't drink it because of the salt content. A number of people trying to use new types of materials to create filters or other materials that will remove salt from water. The picture you see in the upper right-hand corner is of a thing we call a quantum point contact. These are people firing electrons through a tiny sort of gate to understand how electrons behave in the hopes of making the next generation of computing technology that we think will use what we call the spin of an electron instead of its charge. And this is a picture of a prototype for what we call a lab on a chip. A number of people in a number of places trying to make biological sensors or detectors that can do the kinds of measurements on biological samples that currently take an entire room in something the size of the palm of your hand. Currently, if you have a blood sample or a urine sample and you want to test it for 15 different things, you need to cycle it through 15 different machines. A number of people doing very smart things, trying to make it so that you can do the same number of tests with just a few drops of that same material. And that's the sort of, as I said, real forefront of where nanotechnology is today, where the most interesting questions are being tackled. But it's not the only thing that Feynman originally mentioned, and it's also not the final frontier of where nanotechnology might go. Because Feynman also suggested that we might make a revolution in what he called miniature machinery. And this is where some of the more far-fetched and hard-to-believe suggestions about nanotechnology have come from. And the fear of some of those things, which may or may not be possible, is one of the things that's motivated these types of public outreach and education efforts. And this is the type of science fiction image that you can find if you surf around the internet looking for information about nanotechnology. We have here some presumably glass or plastic robot grabbing onto a red blood cell to inject it with some fluid of unknown origin or purpose. And there's a number of reasons why this is either impossible or a completely useless thing to do. The first is, based on whatever that object is designed to be made of, you simply couldn't make something that fine a scale. If it were glass or plastic, it would be impossible to manufacture it with that kind of precision, and it probably wouldn't be rigid the way you'd want it to. The second is that I can't think of any substance that you would want to inject into a red blood cell. Red blood cells don't have DNA. All they do is carry oxygen. I can't imagine what you would be treating it for. The third is that I can't imagine how many cells this device can treat itself before it runs out of that fluid. So you'd have to add enough of these devices comparable to the number of red blood cells you have. I don't think anyone would want a gallon of those things injected into their bloodstream. This probably wouldn't work. But that doesn't mean that there aren't things on this size scale that we could do that would be interesting in, for example, biology. This is an artist's conception of a tiny machine that actually exists. This is a T4 bacteriophage, and it's in fact smaller than the device I just showed you. It's a virus, and it's a virus in particular that eats the E. coli bacteria. It lands on E. coli, injects a strand of its DNA, creates more viruses until eventually the bacteria ruptures and the virus run around and find more E. coli to eat and kill. Now that's a pretty interesting and complex system. We didn't build it, it evolved on its own, so it's not technically a machine, but there's a lot that we can learn about how that system works, and the day may come when we can in fact redesign systems that work at that size scale and with that level of sophistication. And it's important to remember that biology knows how to do all kinds of really interesting tricks that we know nothing about or that we absolutely cannot recreate. A pine cone contains a number of seeds that have on them all of the information, all of the engineering diagrams to build a giant sequoia tree. And the raw material to make that tree comes out of thin air. The tree is pulling carbon dioxide directly out of the atmosphere, using the sun's energy to bond those carbon atoms into big molecules to create the material that's in the tree. All the information is contained in the seed, and all you need to do is put it into the soil, and it somehow knows to draw the nutrients and the raw materials that it needs to create that gigantic system. This is not a designed system, it's an evolved system, but it does these incredibly complex things, and we are beginning to understand more and more about the machinery that runs these kinds of systems, and that may enable us to recreate systems that work with something like this level of complexity. Because it's important to remember that the encoding for those systems, and indeed for systems like us, is largely an information processing question. You can think of DNA as a code, not at all dissimilar from computer code. And one of the things that's interesting about nanotechnology is that for the first time, people from fields as disparate as genetic engineering and computer science are talking to one another and seeing if they can't share ideas and methods. It really is a science in its infancy. So I would argue that there's a place here where the three traditional branches of science sort of all coalesce and come together. That place is the place where we're doing nanotechnology. It really is a convergence of these three sciences. People working in labs that are doing nanotechnology are bringing very disparate scientific professionals together to work on the same problem. It's not uncommon to find a biochemist and an electrical engineer working together to achieve a specific goal. I want to thank you very much for your time and for your attention and for coming here this evening. Thank you.